that my research became a bit more diverse in the last couple of years after establishing my own group. But one of the topics that I've been investigating, I would say since 2014, is Coccolithophore biomineralisation, which is a really interesting organism in itself, also from of course, the mineral formation point of view. So, for those of you who are not familiar with Coccolithophores, so these are marine unicellular algae. We see here some scanning electron microscopy images. And the reason I'm interested in them, and many people are, is first of all because they produce these scales of calcium carbonate, where each scale is essentially an assembly of individual crystals of calcium carbonate in the form of calcite. And one of the main interests in the biomineralization community is understanding how organisms do that, and that's also for me the main point. But it's not just that. They're pretty important organisms for the geochemical cycle in the oceans. I mean, they form the largest geological sink for carbon, and they contribute to the transport of matter to the deep ocean. So they are one of the most prolific calcifiers in the ocean. What happens is that when they die, they sink to the bottom of the ocean, where they carry all that organic matter. And of course, the coccolids are very important to provide ballast for them to sink. And what you see here is a satellite image of a bloom of Coccolithophores on the coast of Cornwall. So they're known for making these huge blooms in the oceans. And the reason we can see them from space is because of the calcium carbonate, and all that white cloud that you see here is the Coccolithophores. And that's essentially looking at the calcium carbonate crystals that refract the light and make them visible. So they're quite important for the ocean. The calcification that they do is very important for the geochemical cycle. And we still know very little about how they make those crystals and why they calcify, why they produce those scales. I mean, as my previous postdoc used to say, nature doesn't do things for fun. So if they're investing energy in calcification, does cost energy, and it's not a random process, which you can see in the structures, that it must serve a purpose. But there are many hypotheses why they do this. We still don't know what is what. Another fascinating point about coccolids is when it comes to the diversity. So what we're looking here is 6 different species of Coccolithophores, and this is just a small example of the variety of different species that calcify. And what you can notice is that each one has scales with completely different morphologies. And these morphologies, they are species specific. So that points to the very intricate biological control that these cells are exerting over the formation of calcium carbonate. So all Emiliania Huxley, they're going to produce coccolids that are going to be all roughly in this shape. Sarcosphaera absteni is going to make these coccolids of these cups and so on. So it's again brings to mind how are they controlling the calcification of calcium carbonate, and of course everything is done. It's biologically controlled, so it's all encrypted at the end of the day in the genetics of each organism. And here I just wanted to emphasize the morphological control over calcium carbonate formation in coccolids, which I think for me, it's what drew me into investigating them, because I really find this, looking at these images, I really find this mind-blowing. So what we see here on the right is a synthetic crystal of calcite. This is the kind of calcium carbonate crystal that we get if we just mix in the lab calcium chloride with sodium carbonate or bicarbonate. So we get these rhombohedral-shaped crystals. After a couple of days, they're going to be several tens of microns in size. They have this very regular structure. It follows the unit cell of calcite. So that's how calcite likes to form if we leave it on its own. Now, if you see here on the left side, this is what coccolids from Emiliania Huxley look like. I mean, they have each one of these structs is one crystal. That would be the equivalent of one of these. They have this very elongated shape, very high aspect ratio. We see that they are a few hundreds, about 600 nanometers in length. They are tens of nanometers in thickness. So they're completely different from what calcite would normally form in the lab. And that is again emphasizing the remarkable control that the cells can exert over the crystallization of calcium carbonate. It's controlling the size of the crystals, the shape, and of course the assembly of different crystals into this kind of structure. And understanding how they do this is one of the questions that I'm after, and many people in the biomineralization community or after, not just from coccolithophores, but people who investigate bone formation, coral skeletons, mollusks, and you name it, and you find at least one group working on these. This is an overview about how coccoliths calcify. I mean, we know quite a bit about what they do by now. And this is a scheme of a cell. And what is important to notice here is that, first of all, calcification takes place into this compartment here that is called the coccolith vesicle. And we have the involvement of essentially two sets of macromolecules. One is what we call water-insoluble macromolecules. These are essentially a scaffold or a substrate for the crystals to nucleate and grow. And on coccoliths, these are called the base plates. So I'm going to talk about these. And we have water-soluble macromolecules, and these are essentially polysaccharides. We call them coccolith-associated polysaccharides. They tend to be highly acidic, and they participate in different aspects of mineral formation. We don't know exactly what they do, but there is no evidence that they assist with transporting the calcium to the base plate for the calcium carbonate to nucleate. They may be involved in templating the morphology of the crystals, or also in selecting the type, the structure of calcium carbonate that is going to form. If I have time at the end, I'm going to talk a little bit about these. And then there is a compartment. Actually, I think that there is more than one compartment that serves as calcium reservoir, where the cells up the calcium from the extracellular medium, from the seawater, basically, it goes into the compartment. We don't know exactly how. We know that there are some calcium pumps that do that, and then the calcium is transported to the coccolith vesicle. We don't know how that happens. So this is a general mechanism, or general overview of the mechanism of calcification. Of course, the devil is always in the details, and we don't know in detail how all these processes operate. And these, again, just to emphasize 4 different species of coccolithophores producing structures with completely different morphologies. So what I've been doing the last years is to use a number of cryoimaging techniques, and it's mainly cryotransmission electron microscopy and cryogenic tachography, to try to characterize different steps or different stages in this process. And working under cryogenic conditions is very important, because these are, of course, we're looking at cells here, which are biological structures. They're hydrated. I mean, cells contains huge amounts of water. And if we want to study how coccoliths are made, how are the cells doing the development of macromolecules, we have to make sure that whatever imaging technique we use, we can preserve the cells and any kind of macromolecule that we want to observe in their state, hydrated state, because otherwise, if you stick into a normal microscope, high vacuum will require that the sample is dried, and then of course you remove the water, things shrink, morphologies change, and then we cannot be 100% sure of what you're looking at anymore. So that's actually, I would say that it's the key thing in here, is to be able to look at these samples under cryogenic conditions. So this is an example of a cryo-TM image of what we call a proto-coccolith ring. So this is a coccolith that was done by my former postdoc Bart Marzek, that we managed to extract it from the cell, freeze it, and image it using cryo-electron transmission microscopy. And what you see here is the very early stage of the formation of coccolith ring, which already points to the question of how are the cells assembling single crystals into this kind of a ring or a scale, a polycrystalline structure. And we see that it's actually, in a way, simpler than what you'd think, because what the cells do is to simply nucleate individual crystals of calcite in a kind of a ring. And then these crystals are going to grow selectively along different directions to reach the full morphology of a coccolith. Of course, again, how the mechanisms controlling these processes, we don't know. The first point that I'm going to talk about is control over crystal nucleation, and I'm going to be focusing mainly on this species here, which is Pleurochrysis cartera. Now I think that the name changed to Chrysotila cartera. I still can't get used to the new name, so I keep calling it Pleurochrysis. This is an image of the cell. If it looks a little bit or quite a lot deflated, this is what happens when you run it through a filter it using vacuum and stick it into the scanning electron microscope. The cell is just going to collapse. But we weren't interested in the cells. We just wanted to get an image of the coccolith on the outside, which you can see here. And this is just a scheme showing how the crystals are organized into this different structures. So we have types of crystals. They both have similar morphologies. They call it NU shaped. We have the V units, which means that I have the crystallographic C-axis vertical to the direction of the coccolith. So these are basically standing up, as you see here. And the OR units, which are interlocked with the V unit. We see that here. And the crystallographic C-axis is basically radial in the direction of the coccolith. So we just have this sequence of V and R crystals alternating and mechanically interlocked, which gives stability. And of course, what I need to highlight here is that they grow on an organic substrate that I explained in the beginning, which is the base plate, the coccolith base plate, which here we cannot see, but it is here on this scheme here. Base plate is where everything happens. So that's where the crystals are going to nucleate and grow. This is a cryo-transmission electron microscopy image of a base plate. We don't see a lot, to be honest. I mean, we see that it has an oval shape, but we don't see much structure in there. That's because it's a substrate that is very, very thin. We're talking about 20 nanometers in thickness. This is an atomic force microscopy image from a more recent paper that again shows how the base plate looks like. And this is the old work of Mary Marsh, where we can see calcium polysaccharide particles being delivered to the rim of the base plate, where they are starting to form a ring of crystals, which later develop again, and this ring of alternating rhombohedral crystals, alternating crystallographic orientations, and then they're going to grow to form the mature structure. Of course, the big question here is that if we have calcium carbonate nucleating around the rim and not in the center of the base plate, that means that something must be different around the rim that is providing nucleation sites, and that must be absent from what is inside. So there's a model that again was proposed by Mary Marsh. Jeremy Young talks about this as well, that there's a kind of what they call a folded ribbon template, which is a macromolecular or a template on the rim of the base plate that is providing nucleation sites with alternating crystallographic orientation. So you can think of it as a checkerboard array of nucleation sites on a folded organic substrate on top of the base plate. Back in 2016, Asaf Gal did some very interesting work where they showed that acidic polysaccharides, with coccolith-associated polysaccharides, they direct calcium ions to the rim of the base plate, forming this kind of cloudy material in here. And that is specifically on the rim. So again, nothing is binding to the inside of the base plate. They call this a macromolecular recognition mechanism, meaning that the acidic polysaccharides somehow recognize the rim of the base plate and bind there, bringing the calcium ions. And that raised the questions for us, what is the mechanism there? Because if you're talking about macromolecular recognition or whatever mechanism is, it means that the rim has to be chemically different from the inside of the base plate, and to enable the binding, the specific binding of the polysaccharides by whatever mechanism. That's what we set out to do, to try to understand what is driving this. And the first thing that we did was Cryelectrotomography, which means that it's a technique to do imaging in 3D using the transmission electron microscope. If anyone wants to hear more, I can explain. And we did that on the base plate, and we could see that the base plate actually has a much more complex structure than it looks like just from this to the image. I mean, it has this kind of a 3-layer structure that is comprised of of fibers of different thickness. What is in green here, which I'll call it bottom, is made of cellulose fibers. It's very organized, but we don't know yet what these other disordered structures are made of. What is interesting here, though, is that the rim of the base plate in red, which is where the crystals nucleate and grow, is morphologically different from what is inside. So that is already showing that there is a difference in here in morphology. Now, we did some experiments using polymers as mimics to the acidic polysaccharides, and we could see, this is another cryo-TEM image, that when you mix polyacrylic acid with calcium chloride, now we're using polyacrylic acid, it's highly acidic. Polysaccharides are also highly acidic. The Coccolithophore polysaccharides are highly acidic, so it's a suitable mimic in that sense. And it forms complexes with calcium ions, and then it binds to the rim of the base plate. And what this is telling us is that if we take a polymer, that the only thing it has in common with the polysaccharide is the charge, the amount of charge residues, and we can get it to bind to the rim of the base plate, it means that the interaction is not specific, that it's something else. And that something else must be simply electrostatic interactions. So we have a highly negatively charged complex, even in the presence of calcium ions. And that means that the rim of the base plate must be positively charged. Because this is a biological substrate, any positive charges would be very likely be deriving from primary amines, could be from aminated polysaccharides or amino acids like lysine. So what we did to test that was to use a fluorescent dye, Echography, that is functionalized with the 10-hydroxysuccinamide residue, which makes it to complex specifically with primary amines that are present on a substrate. And this is a fluorescent image resulting from this. And we can see that the staining is not perfect, but it's mostly localized around the rim of the base plate. What this is suggesting is that the rim of the base plate is functionalized with primary amines that are going to be positively charged, and they're going to be providing an electrostatically privileged binding site that can attract or bind to negatively charged complexes of polysaccharides and calcium ions. So that means that we don't need any specific macromolecular recognition, as it was called then. All we need is favorable electrostatic interactions between the polysaccharide and the nucleation sites on the base plate. And that can be enough to drive the polysaccharides and the calcium ions to the nucleation site. That is essentially the first clue, the chemical composition of the nucleation sites on the base plate. So that is already giving us a clue of what's happening here. Whether in biology it's purely electrostatic interactions or there are other mechanisms in place, that's something that we have to further investigate. Now I'm going to talk a little bit about control over crystal morphology. And this is continuing on Pleurochrysis cartera, and just to remind you how the crystals look like. What we did here was to extract coccoliths from the cells by bursting the cells open and freezing them with just cryopreserving them and imaging again using cryo-TEM. This was the work of my former PhD student, Jessica Walker. And then what we did was, again, cryoelectrotomography to image them in 3D. So I have here two coccoliths at different stages. This is very much still forming. This is an almost mature state, but not quite there. They came from inside the cell. And this is how the 3D reconstruction looks like. Sorry. We have here the crystals. We can see the base plate here in red. We have here some blue spherical particles at the top and some brown ones at the bottom. These are the coccolithophones, which were calcium polysaccharide complexes that somehow survived the extraction process and remained attached to the crystals. And we have here two forming V and OR units, so two different units, different crystallographic orientations. They are mechanically interlocked. They're not fully formed yet because you can see that this one the units in green and this kind of pink color still have to grow more upwards and form this kind of anvil-like structure. And the one in the middle, it's starting to grow the coccolithophore from the back. And this is the late stage. It's all almost mature. And we have these coccolets here that already have the morphologies, the VN or unit, and how they interlock. Now, luckily for us, coccolith formation is not perfect. Sometimes there are defects or mismatches. And that's where we can learn when things go wrong, we can use it to learn what is it that makes things go right. And if we pay attention to this coccolith, we have here a region where we have two ore units that are adjacent to it and right next to each other. So basically we have a V unit missing here from the middle as we have in this part. And you can see that the morphology of the crystals is very different. So they're not as limited in terms of growth. They're just growing without any restrictions to the top and to the bottom, which is indicating here that the presence of a V unit here in the middle would be very important to restrict the growth and basically bring this anvil kind of morphology. And you can see the same thing in a region where we have three V units that are adjacent, where what we're missing, especially if you look at this blue crystal in the middle here, instead of having these indentations here in the middle from kind of a T shape, it just right flat on the side, and that is because we don't have an R unit growing next to it that would be basically restricting the growth of the crystal to the side. If you look at the back, where normally this crystal is going to be very thin at the back, I can show it here. It's very thin here at the back. It's just a uniform thickness because you are lacking a part of our crystal growing from behind that is restricting the growth of this crystal backwards towards the outside of the coccolith ring. And same here, same thing in this purple one here. So what this is basically telling us is that one of the mechanisms for controlling morphology, it's a matter of competition for space. So we have crystals that are growing in a crowded environment. They have different crystallographic orientation, so they're going to be going faster along different directions. And by doing that, they pin the growth of the crystals that are just adjacent to them. So they're restricting the growth of that crystal, and that is helping to give rise of this kind of morphology that you observe. So it's a matter of competition for space, which again means that the process doesn't have to be very complicated to template the morphology. Of course, that may not be the only mechanism at play. There may be other factors affecting. We cannot discard the effect of the polysaccharides. The coccolith vesicle, it's probably also delimiting the growth of the crystals, but it's also a matter of confinement and growing crystals in a crowded environment, and they will do it themselves by restricting their growth. I'll now talk about a bit about echography, and this is work that is more recent. It's the work of my PhD student Alex Trikas, and so we're looking here at the development of the mineral inside a coccolith. Now, echography, it's a kind of an interesting and a really exciting imaging technique. It's been gaining traction, I think, in the last years. It underwent quite a lot of development. And just to talk briefly what it is, it's a form of imaging technique using X-rays. So in a way, you can think of it analogous to X-ray tomography. So we have a sample, we fire a beam of X-rays there. We have to do that in a synchrotron. But instead of getting an image like you do X-ray tomography, or even if you make an analogy to TM tomography, what you get is a coherent diffraction pattern of the image. And what we do is that we raster this image, the sample with the beam, to collect overlapping diffraction patterns. And then, of course, we rotate the sample to do that in 3D. So it's a process that actually takes several hours to acquire a data set. And then through some black magic algorithm that I won't pretend that I completely understand, we can reconstruct what the sample looks like. And it has two advantages over X-ray tomography, which I think makes this technique really exciting and really useful. One is that the resolution is better. So we can get resolution in the order of 50 nanometers, sometimes a bit better than that. And the second is that it's quantitative. So we get an electron density map that we can directly correlate it to the composition of the material. So these are examples of data sets that we collected from Emiliana Huxley here on the left and crisis criteria on the right. This is a slice from the 3D volume, so just looking at the cross-section from the cell. We can see on the outside mature cockpits. This here circled in black is a compartment, and it's brighter because it contains calcium ions. And we see here circled in red a formed coccolith. And this is the 3D rendering that we did with the computer. This is just showing the cell from the outside. We only see the mature coccolith. Here on the inside, we can see a formed coccolith in pink and some intracellular compartments in black. So we can get basically a 3D reconstruction of the whole cell, showing all its contents. And this is a pure crisis. So it's the same thing. This very thick white border is just a glass capillary, so don't mind that. We have two cells that are squeezed inside, but we can see here quite a lot of intracellular compartments, and we can see here forming coccoliths as well. And this is reconstruction of just the intracellular compartment inside the pleurocrisis cell. So we can get quite a lot of information out of this technique. So I'm going to focus now only on Emiliana. I won't talk about pleurocrisis, but the data is actually very similar. So looking at, so the way that they made the cells is that we had to load them into a glass capillary, freeze them using a plunge freezer, and then they are analyzed frozen at the synchrotron. So everything here is cryopreserved. And in the capillary, we just have a very large number of cells and have the 3D reconstruction of all of them. So basically, it enables us also to do some statistics, actually. And by looking at the cells and the reconstructions, we could pinpoint coccoliths at different stages of formation. I have highlighted here 4 stages from 4 different cells. It's interesting that we have coccoliths being produced at the same time on Emiliana. And this is the reconstruction from 4 different coccoliths from each of these stages that are representative of what we observed. And we can see very much matches what is in the literature. We have the formation of this proto-coccolith ring, as it is called. It's a ring of rhombohedral crystals of calcite. We don't have the resolution here to resolve the crystal, so that's not why it looks like a continuous ring. The next stage, we see that the proto-coccolith is larger, is taller, so it's what we call the kind of a 2-layered structure. It's taller, and then we start to see growth towards this inner part and the start of development of those struts of the coccoliths, and then we get mature coccoliths, still intracellular, but they already have the mature morphology, and then you have the extracellular ones that are here in yellow. This is all very interesting and very exciting that we can pinpoint these stages, but there's more to it that we can extract. I mean, I mentioned that what is really exciting about echography is that it's quantitative. So we can get an electron density value that we can directly correlate to the composition of the material that we're looking at. And we can do the segmentation, the computer reconstruction based on electron density that we see in the coccolith. And just to give you a reference, the coccoliths are made of calcite. It's calcium carbonate in the form of calcite. And the electron density that we expect for pure calcite, and this is both calculated and it's also roughly what is observed on materials like mollusk shells, which is also made of calcite, is that we'd expect to see values of about 0.82 electrons per square angstrom. And this is the part that was really surprising for us, because when we did the segmentation based on the electron density, we got something else completely. these are 5 stages or 6 stages of formation that is based on the electron density. So here you have the morphology of the coccolith at different stages, from proto-coccolith ring to when you start growing the distal and proximal elements, so these struts, all the way to micrococcolith. And what is really striking is that if you look at this early stage here, we have a ring that is presumably of calcium carbonate, but it's very low in electron density. We're talking about electron density between 0.4, mostly in the ballpark of 0.5 electrons per square angstrom, with very few regions a little bit over that, but not going over 0.62. When you look into the next stage, we start seeing voxels or areas of the coccolith that are higher in electron density. So that means that there is some maturation process in the mineral. We can discuss that a bit more, what that means. And when we look into the following stage, which is when I have this 2-layered coccolith, we have electron density between 0.72 to 0.82. But here I need to point out that in this range, it's mostly still within 0.72. And this is forming the core of the coccolith. Most of it is in the region between 0.62, 72, or 0.5 to 62. That is, you can think of it as an onion, with the inner part being higher in electron density and then forming a gradient going lower in the electron density as you go towards the outside. And indeed, we see some very low electron density regions going towards the outside, which could be indicative of amounts of organic that is coating or getting occluded in the coccolith. And from this point on, nothing much changes when we think about this high electron density region. We end up with a crystal that, a coccolith basically, that has a core that has electron density that is approaching that of calcite. It only forms the core of the structure and only the center of the ring, and everything around it will start going lower and lower and lower in electron density, and especially when it comes to the calcium carbonate, to the crystal struts that are pointing out. So that means here 2 things. I mean, one is that there has to be some kind of maturation process or changes in composition that is happening. And what I mean to say here is that the coccolith is not purely calcite, which is very surprising, because we know from the literature that the amount of organic polysaccharides that are included in the Emiliania is a few percent. So talk about probably 2 to 5%. So it's not enough to explain these differences. But there is something happening when you're going from this stage to this stage here, which may indicate that at the very early stages, what you have is not calcite, but it's some form of calcium carbonate that is lower in density. And that could be amorphous calcium carbonate. It could be very, very hydrated mineral. And then it has to crystallize. It's going to extrude water. It's going to probably, it may extrude polysaccharides or other organic material if they're in there, and then they're going to increase in density. So there's some kind of a maturation process going on here. And that happens mainly at the center of the coccolith, and after that, it stays the same. And that is indicating the remaining of the structure, the outer layers, if you will, it's either, again, containing high amounts of organic, or maybe it's a porous structure. That is something that we're still working out how to explain this exactly. But there is something really interesting and really thought-provoking about that's happening here. But the point here is that they are made of calcite. Each strut is a single crystal of calcite, but maybe this is not really pure calcite. There is something else in that is contributing to this very low electron density. To check for that, we did some other measurements on mature coccoliths. One of them was nanodiffraction, and this is just to confirm that the coccoliths are single crystalline calcite, and this is really what we see. It's clear it is a diffraction pattern taken from a different region in here, and that follows pretty much what is known in the literature. Each one is a single crystal of calcite, so nothing much to see that. But what was really interesting is that we also did quantitative energy dispersive X-ray spectroscopy on these to quantify the amount of calcium, the calcium to oxygen ratio. Now, for calcium carbonate, we'd expect to see a calcium to oxygen ratio that is about 0.33 or something like that, 0.35. And this is what the scale here is showing. And this is what we get in the central ring of the coccolith. So the area of the coccolith that is highest in electron density, if you remember this electron density segmentation, is the same area that has a calcium to oxygen ratio that is consistent with calcium carbonate. As we go outwards, the other to the distant proximal element and away from the central ring, we see that the calcium to oxygen ratio decreases, which means that if you have more oxygen than calcium, very possibly we're talking about higher organic content in here compared to the central area. So that could explain the discrepancy that we see in electron density and the differences between two areas. And it means that the composition of the coccolith, first of all, is not homogeneous throughout the calcium carbonate crystal. So I have regions here that contain a higher calcium to oxygen ratio, you can say higher mineral content, and areas that contain lower mineral content, a higher organic content. So that is a take-home message in here, and that matches well with the tachography at least. And then if I still have a couple of minutes, I'll try to be quick on this part, which is just talk a little bit about control over polymer type. Now, this is a study that again was done by Jessica during her PhD, which is studying the function of these coccolith-associated polysaccharides in controlling mineral formation. And we know a little bit about what they do in terms of controlling morphology. I mean, it's been shown in the literature that they can promote the formation of crystals that are elongated in a way that resembles the coccoliths, the single crystals in the coccoliths. So we can see here in this image, I have this crystal that instead of a rhombohedral crystal is very much elongated. And that is because it's making the crystals grow faster along the C-axis. The question that we're interested in answer is whether these polysaccharides can promote nucleation and whether they control the polymorph type. So calcium carbonate can grow into 3 different polymorphs, which means 3 different materials of different crystal structures. Calcite, which is the most stable one, and is what we found in coccoliths, but it can also form Aragonite, which has a different crystal structure, it's slightly less stable, and vaterite, which is the least stable one. And we're interested in asking whether these polysaccharides can direct the formation of calcite towards, even in conditions that would favor the formation of other polymers. So what Jessica did was relatively simple. We got polysaccharides extracted from the coccoliths. We put them in a supersaturated calcium carbonate solution under conditions that would favor the formation of vaterite, and then collected samples over time and froze them and imaged using cryo-TM. And this is the images we got. So this was done on Capsa oceanica, so it's a different species. And this is the control without any additives. And what you observed is the formation of these spherical particles. So this is amorphous calcium carbonate. It's not crystalline yet. It's known to form as a precursor to calcite. And then over time, it crystallizes into vaterite and calcite, which is what we expected. And the proportion between the two is pretty much 50-50. When we added polysaccharide in the solution, what we had was very quickly the formation of calcite crystals, and only calcite crystals. 100% of the material that we got out of this was calcite, even though we were under conditions that was favoring the formation of vaterite as well. We also checked conditions that would favor the formation of aragonite, and that's what you see here. And this is again the control without additives, and we follow the protocol that is in the literature developed by Karin Sand and Susan Stitt. And the result was very much consistent without additives, without polysaccharides. After 1 hour, we get essentially aragonite, which is a different crystal structure. But adding the polysaccharide, we ended up with a good proportion of calcite, which you see here. These rhombohedral crystals are all calcite together with aragonite. So what this is essentially showing is that the polysaccharides, they have the ability, at least in Jafar capsa, to tailor the polymorph of calcium carbonate that is being produced. And this raises an interesting question about the chemistry in the Coccolith vesicle. So we know very little about the physical chemical conditions in there and whether those conditions were enough to produce calcite, but it could be the case that without the polysaccharides, you would get a different polymorph. For example, if there's enough magnesium in there, you're going to tilt it towards calcite, but the polysaccharides are going to be tilting the reaction towards calcite. So it raises an interesting aspect to the possible function of these polysaccharides. So I'm going to conclude. So the main messages, main things that we learn is that, first of all, about the base plates that have these very complex structures that are composed of 3 layers. We have the rim where crystal nucleation occurs. Is morphologically and chemically different from the inner part. And the difference in chemistry is this functionalization with positively charged amine groups that form binding sites for the polysaccharides to bring calcium ions. In terms of crystal growth, we can see that it's constrained by space due to the presence of adjacent crystals, and of course that is going to form an important mechanism to control morphology. With echography, we could see the morphological development of the mineral, and that indicates that there is some kind of maturation process going on there, perhaps through a disordered precursor, a highly hydrated precursor. It's also showing that the composition is not purely calcite. We're still trying to understand what is happening there. And the polysaccharides that promote calcite nucleation, even under conditions that favor other polymorphs. And that again could be raise interesting questions about the chemistry in the vesicle and of course the function of these polysaccharides. Last, I want to thank my group members, present and former, especially the people who did the work. So Alex Trikas, Bart Mazek, and Jessica Walker, all my collaborators and funding agencies. And thank you all for your attention.
Thank you, Fabio. That was a really interesting talk. So we're going to open up for any questions that you may have. So either put them in the chat or raise your hand, and we'll go around asking some questions. So has anybody got any questions? Ooh. I think so. Right. Oh, that was my bad. Yeah. So yes, Jeremy, you got a question? I think you're still muted on your end for some reason.
I think so. Can you hear me?
It's very, very quiet.
Quiet. Maybe a microphone problem. I'll try talking loudly. Can you hear me?
Yep. Just if I turn my microphone really, turn my speakers up.
Sorry, not sure what the problem is there. Yeah, fascinating talk. Lots of lovely stuff in there. I very much agree with you about that space competition as a fundamental control on the crystal morphology we see. I've always thought it'd be a fascinating thing to try to get someone to do computer modeling and to actually just start with the hypothesis that the biomineralisation controls first the nucleation direction and then the space the crystals grow into, and you need very little other control to make a lot of the complex internal form. I think it'd be a fascinating, maybe an interesting way to go. The part which I guess I most wondered about in your talk when you're saying this thing about having impure calcite, the evidence for that, and I wonder whether you can't explain those observations as just varying ratio of organic coating to coccolith, to have pure calcite but with a lab organic coating on it, and just as you go outwards from the center, you've got less calcite and more organic coating.
Yeah, I think that I mean, in terms of computer modeling, I completely agree. I think that is going to be extremely interesting to track. About your second question, I mean, you touch on a point that I mean, it's the kind of thing that I've been wondering ourselves. And I mean, what you mentioned about organic coating causing the decrease in electron density, that is very much true. And this is what we refer to as partial volume effect, which means that We have essentially a voxel, which is, you can think of it as a pixel in 3D. You have both mineral and organic in there. And of course, the resolution of the technique puts them both in the same box, and you end up with a lower electron density. And I think that this is very much what is happening when we look at the proximal and distal elements in the coccolith, which are these struts, because they are predominantly very low in electron density. And of course, that is going to be a mixture of occluded polysaccharide and the coating of the polysaccharide on the surface. I mean, we're talking about structures that are a few tens of nanometers in thickness. So that is very much what's happening here. What is happening on the inside, it's a different matter, because here the coating of the polysaccharide is on the outside of the coccolith, and that is far enough away from the core of the coccolith to cause this decrease in electron density that we see here more in the center. So it cannot explain what we see there. So it has to be something else in there. That's all I can comment on. I mean, from what people talk about in the literature is that you have polysaccharides coming in mostly once the ring of calcite has nucleated. So that would have been once we have this kind of structure in here, and then the crystals start growing in different directions. That's when more polysaccharides start coming in. So it could explain why we started seeing this lower electron density. But again, if you go back to how much polysaccharide is actually in there, that's what I find challenging, because you need quite a lot of polysaccharide to cause this decrease in electron density, basically.
Awesome, thank you. So we've got a few questions in chat, so we'll just go through them and then we'll go back to see if anyone's got any other questions. So firstly, we got wondering whether you think it would be possible to use these methods to investigate hollow coccoliths formation.
I think definitely. I mean, picography is a very powerful technique. We can see that both inside and outside the cell, and I think that is definitely possible. There are other techniques that can be used, like cryofocus ion beam scanning electron microscopy. It's another very powerful technique to do that. Cryo-TM is more challenging because you're limited by a sample thickness. So we cannot have any sample in there that is thicker than 200 nanometers. So we have to be very specific about what to look for. I mean, essentially, you cannot put a whole cell in there. So for that, we have to look at techniques for like cryofib or tachography, which would be really exciting to do. Then you can see what's happening inside the cell during calcification.
Of course, yeah. Another question we got: any idea what the base plate is made of, and what is the carbohydrate?
So the base plate is cellulose that forms the backbone, and then the proteins. What these proteins are, I don't know. I'm not sure anyone knows what they are. I think people are still trying to purify and identify these. I think that the group of Andre Scheffel has been working on these. That's essentially all that we know about the base plate. And in terms of what is giving rise to the primary means, I mean, it could be proteins, so have lysines, for example, or it could be a polysaccharide, but I don't know. The coccolith-associated polysaccharides, they are rich in uronic acids. So I talk about mostly galacturonic acids, and that is what is giving the charge.
Awesome. So we've got one more question in a minute in the chat, and it's quite a long one. So in this SLIBN model, they show two compartments, one that stores calcium first and one where coccoliths form. Have you seen anything consistent with that in your Tycho analysis or any other analysis? What would be the form of calcium in the first compartment, and how would they exchange calcium so that there is plenty of calcium in the second compartment?
So this is one of the questions that we're trying to address. I mean, we have data. Let me share my screen again. Let me go back. So we have the data. I mean, we have all these 3D images from the cells, and they obviously they show all the compartments inside, especially obvious in pleural crisis. And because calcium-containing compartments are going to be higher in electron density, we can pinpoint them because they're going to be brighter. We haven't fully analyzed the data yet because we've been focusing more on looking at the coccolith formation itself, and that has been on its own already enough work. But what we can say, you can say a couple of things here. One is that it's not just one calcium-containing compartment. Actually, there are several compartments in there. You can see here, I think that if I remember correctly what Alex did, I think that all these magenta compartments, they contain calcium, and some of these blue ones. Sometimes you have calcium-containing compartments inside all other compartments. So it's quite complex, the picture, in fact. So it's not just one, it's several. Sometimes they appear in touch with the coccolith vesicle, or at least in very close contact. Sometimes they are far away. So it's not entirely clear yet what is going on, and especially how the calcium is being translocated from these to the coccolith vesicle. I mean, Alex is working on this. He's been doing also some live cell, some confocal imaging, tracking the calcium. And I hope to be able to answer this question very soon. But at the moment, that's all I can say for now.
That's good, thank you. Has anyone else got any more questions in the audience? If anyone's typing, there is one question that was from the registration, actually. So it's, how is or would calcification be affected by acidification?
Yeah, that's another good point, and it's something that people have been looking at. So I don't think that the picture is very clear yet, and I think one of the reasons is because the calcification happens intracellularly, and the cells are very good at regulating the composition of the compartment. So as long as they can maintain the influx of and the equilibrium of carbonate and bicarbonate in the vesicle, and as long as that is not affected by the acidification of the seawater, then the acidification shouldn't affect very much the calcification itself. Of course, if you go to an extreme where that becomes harder to pump carbonate or bicarbonate to change the equilibrium, that can change things. But I think that they're going to be pretty resilient to some extent to ocean acidification.
Awesome, thank you. So I don't think there are any more questions. So again, thank you very much, Fabio, for this. It was a really interesting talk, and thank you everyone for joining us today. Alex, want to introduce next month?
Yes, so next month there is a slide